Thank you very much, Mark, for that generous introduction, and good morning, everybody. Glad to be here from Nigeria, and I have warm greetings from colleagues in Ibadan and from Ihendri and colleagues in Indianapolis. If we go to the previous one. Yes, so we have the logos of the University of Ibadan and University of Indiana. And I'm glad that I'm the first to speak so that I can dispense with the soft science and then concentrate on the harder stuff coming on later. I have nothing to disclose, and I'd like to thank Mark, especially for this introduction and for the hospitality I've enjoyed, but more importantly for his work on dementia. He has shown community engagement in practice, and we have a lot to learn from you. Thank you so much, Mark, for all what you're doing, and we hope we'll continue to work together. For this talk, because time is against me, and I don't want Susan to raise the red flag quickly, I'm going to talk about the Indianapolis-Ibadan study, findings from observational studies, some biochemical assessments, introduction to some genetic studies, then preliminary data from work we're doing currently called the Newark Nigerian study. Then I'll conclude and then make some acknowledgements. Well, we'll start by talking about cross-cultural studies, and they're very important because of many reasons. We have wider diversity of environmental exposures, climate, diet, exposure to pathogens, cultural factors that lead to epigenetic adaptations. Well, we also have chance of identifying unique risk factors, and we can explore gene-environment interactions. And this is the basis, the hypothesis of the Indianapolis-Ibadan study, which was proposed by Oshintokun, that identification of populations or communities with significantly lower or higher prevalence rates of Alzheimer's disease could greatly enhance the search for environmental risk factors for AD. And we have you can see the movement of people during the slave trade from Africa, mainly to North America, and then the offsprings are the ones that we studied. And the two people behind it, Oshintoko and Henry, are in the picture down there. And this was a project timeline. Data collection started in 1992 and went on until 2009. So we had about 7 iterations. In all, we had over 4,000 participants in both sites. In between 1993 and 95, we obtained spot blood for ApoE, and then in 1997 we started doing anthropometric measurements, and we went on, and in 2001 we did cohort enrichment because of attrition due to age, and so we added about 800 at each site, and that's what increased the sample size to about 4,000 on each site, and then we carried out 2-stage assessment at each stage. And this was a outstanding result. I've compressed this together. A prevalence of Alzheimer's disease, prevalence of dementia, as well as incidence, all in one data. And you could see that in African Americans, the prevalence and incidence rates were much higher than in the Yoruba. Prevalence was 8.24 in African Americans and about 2.29 in the Yoruba. Incidence was 3.2 in the African Americans and about 1.4 in the Yoruba, and same applied to Alzheimer's disease, which constituted about 80% of the population there. Now, when you have an increased burden of disease, you need to worry about death rates as well. So the next thing to do was to look at mortality, and in this graph you have the mortality figures over a 5-year period in which we looked at the normal versus those who had dementia. You could see that after 6 months, the individuals who developed dementia started dropping off, but there was not much difference between Indianapolis and Ibadan. The risk of dying was higher in Ibadan anyway, but they were both above 2. The hazard ratio was about 2.05 in Indianapolis and 2.83 in Ibadan. So we didn't think this could account for the difference in rates that we observed in these studies. Now, when you have such figures, then you start looking at risk factors. So you look at genes, you look at age-related changes, you look at environmental factors, and other illnesses and comorbidities. So I'm going to focus on the things we did on environment, living experience, education, vascular factors, diet, and genes, ApoE and GWAS. And this is an aerial view in Indianapolis. and Ibadan, just to make it comparative. And you could see that the thing that was striking about the environment was the proportion that lived alone. In Indianapolis, about 53% of the people lived alone, whereas in Ibadan it was 7.4. So there was that difference in living arrangements, which we felt could involve psychosocial stimulation of people. We then went on to look at the involvement score. We derived it from the PACE study in Northeast America, presence of spouse, visits by people, attendance of religious activities, group membership, and recreational involvement. And we scored 1 for each point. We compared data for African Americans and the Yoruba. And this graph shows what happens. Cognitive decline on the y-axis, social involvement on the left. And you could see that in both populations, the greater the social involvement, the less the cognitive decline, and both in Ibadan and Indianapolis. So we felt that social involvement was important in ensuring that the brain functioned well, and that contributed to cognitive reserve, as you may call it. So we then looked at vascular risk factors. These were self-reported rates of frequencies, and we found that vascular factors were significantly more frequent in African Americans than the Yoruba. Blood pressure, diabetes, smoking, as well as stroke. And so we then did some measurements, like I said, blood pressure and cholesterol levels initially, and found that these values were significantly higher in African Americans than Yoruba, which pointed to vascular factors. In subsequent studies with cohort enrichment, we noticed that systolic and diastolic pressures were significantly higher in female than in male. But at this time, about 3/4 of the African Americans were taking antihypertensive agents, whereas this was not the case with the Yoruba. We also looked at lipid fractions. You could see cholesterol, LDL, HDL, triglycerides. LDL was about the same in both populations, but cholesterol, LDL, and triglycerides were significantly higher in African Americans than the Yoruba. Of course, the thing that you then do after that is to look at diet. And so we compared the diet of the Yoruba and African Americans. We did 24-hour dietary recall, and we also did bomb calorimetry so that we determined the rates. Unfortunately, we didn't publish this data, but what we showed was that the Yoruba's diet was rich in carbohydrate. The African American diet was richer in fat. Protein was about the same. And you could think about this in terms of the things that people ate. Sorry about that. So at that time, we felt that canned foods, hamburgers and such things were things that constituted the bulk of African-American diet, whereas the Yoruba diet, and the example of that is there, is heavy carbohydrate with a lot of vegetables and very little protein. And I think this probably contributed to the differences in the frequencies that were reported in our studies. So we then went on to doing more mechanistic studies. So we then looked at disease pathogenesis. And so we could see that at that time, the cause was Alzheimer's disease, and we're interested in amyloid deposition in the brain. And for amyloid deposition, you need to look at oxidative stress, excitotoxicity, inflammation. Of course, you also have tau hyperphosphorylation. And of course, all this results in abnormal amyloid processing at the membrane level, which is the basis of the pathology of Alzheimer's disease. And this graph summarizes what we had. So endothelial dysfunction, there was no difference between the two populations. E-selectin, PI1, one was high in one was low in the other. But homocysteine was about the same, 17.4 in Yoruba and 17.4 in African Americans. Oxidation about the same, CRP as a protein, the same in the two populations. We had differences in insulin resistance and atherogenesis, but methylation process was about the same in the two groups. So this was published by McDigg in 2008. The first one we eliminated was homocysteine. And I just point to the last sentence that increased homocysteine levels were associated with a similar but non-significant increase in dementia risk for both populations, despite significant differences in folate levels. And so we then dispensed with homocysteine, and then we had to look at other things. So this is the interim summary at this stage. Prevalence of Alzheimer's disease and dementia was higher in African Americans than in Yoruba. Social involvement slows cognitive decline in both populations. Vascular risk factors appear to have some role to play in disease pathogenesis, but biomarker studies were inconclusive. Of course, we then had to go on to do genetic studies. And of all the genes at that time, ApoE was the most important one. ApoE is on chromosome 19. It's linked with cholesterol transport in the brain, and it has three alleles, one on each chromosome. So the E4 allele homozygous is the one that we're interested in. And this summary by Rajabli shows a systematic review that Japanese seem to carry the highest risk with ApoE, followed by Europeans, then African Americans, and of course, Africans have the least risk. But despite that, we then looked at ApoE cholesterol in our population. And this was the first thing that we had. This is the risk of developing Alzheimer's disease on the y-axis, and this is cholesterol. And in those without the E4 allele, we reported in two populations that as cholesterol level increased, the risk of Alzheimer's disease increased in both populations. Whereas for those with the E4 allele, there was very little change in that. And there was an interaction about 220 milligrams of cholesterol. Subsequent studies, we then showed that there was significant association of the E4 allele with Alzheimer's disease, even in Yoruba, but in those with the homozygous trait. Whereas those with the heterozygous, that's the single copy, there was no difference between them and those without the E4 allele. So we concluded that E4 allele increased the risk in the Yoruba if it was homozygous, but not the heterozygous variant. And we compared that data with what was obtained in Caucasians and African Americans. This was a paper by Lindsay Farah when he did the analysis in 1997, and showed that in Caucasians, the risk is about 12, whereas in the Yoruba, this is 12. African Americans about 4, in the Yoruba about 3. But for the heterozygous, you can see that it was not, sorry about that, it was not significant in the Yoruba, but significant in the other two populations. So we can then summarize that by saying that ApoE significantly increased the risk of Alzheimer's disease in the Yoruba, but the effect was weaker. The second conclusion is not correct. It's a bit premature. And environmental and genetic interactions are considered to be highly plausible, but further research was needed to explore biological pathways underlying these associations. So we then put up a conceptual note about the various things involved in AD pathogenesis. We had genes. We had the effect of environment. Oh, why am I doing this? We had genes, environment, gene-gene interaction, gene-environment interaction. So we have ApoE4 allele. There could be other genes. We have vascular risk factors. We have to think about diet, then social interaction. Then we think that E4 expression, there could be other genes involved. And we showed the interaction between ApoE4 and lipids. So this was the conceptual framework at that time. And so we then proceeded to do GWAS studies. So we're looking for targeted variants. GWAS is very important for associating specific genetic variations with particular disease through scanning the whole genome from many different people and looking for genetic markers that predict presence of disease. As at that time, there was no GWAS data from African countries generally. And what you look is to look for the Manhattan plot, which will then pick up whether there is disease or not, or any association. And those are the various genes or genetic traits that seem to be involved with Alzheimer's disease. So from our GWAS data, 5 minutes, okay. I'll run through now. I'll be ready. Yeah, so from our GWAS data, we could see that in African Americans, the APSA7 was significant, but not in the Yoruba. Rather, in the Yoruba, we have clustering gene and the solute carrier family 24 genes that seem to be important. Since I'm going to rush, this is the paper by Brankunko, who is part of the Peggy Vance group, looking at the genes involved with Alzheimer's disease. And because Susan is already warning me, so you can see ApoE, you can see my ApoE is there. Then you can look for the other, APSA-C7 is there, clustering is here, and then the solute something that I talked about, SCL24, is somewhere there. So that provides a genetic profile of the disease. And this is a Rajabli paper that I started by talking about, that there are certain loci that seem to add to the resistance or less pathogenesis of this ApoE4 association with Alzheimer's disease. So we then summarize that, and I'll quickly finish. I have 2 minutes or so more. Now, in African Americans, 2 variants were involved. After C7 in the Yoruba, it seems to be something else is involved. So there are other genetic variants that may be involved in pathogenesis of Alzheimer's disease, and that's why this conference is interesting, because I'll be eager to listen to what is latest in the field in this regard. So I'll quickly end up by talking about the latest study, which is cross-validation of risk factors for Alzheimer's disease in African Americans with Yoruba people of West Africa, which is the NN study, Newark Nigerian study. So the aim was to evaluate how variations in physical fitness is correlated with cognitive function and how this association is mediated by various health and lifestyle variables, and to cross-validate the risk factors for AD in African Americans and the Yoruba. And we have a very rich cognitive battery that we've administered, and we have a lot of data that has come up. We also had some measurements and Rutgers generalization test, which Mark will talk about. We collected sputum for genetic studies and for biomarker studies. And the preliminary results, we've enrolled 124 people, mean age 70, the cutoff age was 60. You can see that there is a difference now. Education now, about 90% of them, including about 30% with tertiary education. So we're approaching what we have in Western countries. Previously, you can see ID study, literacy in Baden was about 15.2%. So things have changed. Living arrangement has also changed. You can see widowed and separated about 37.9. So living alone again has increased. Presently working about 43.5%. MMSE score has increased, 24.9, greater than 24, 90% of them. And hypertension was 42.7, and 51% of them on treatment, unlike before, when there was nobody on treatment at that time. And the other vascular risk factors don't seem to be that important. And this is something about midlife personality that we're going to look at, and a few other things that we'll do by various analysis. You can see that 100% of them felt they were organized, helpful, responsible, self-disciplined, sympathetic, and thorough. And only about 10% thought they were reckless. It would be nice to see how these personality traits influence cognitive performance when we do a further analysis. So I think I'll conclude by saying that dementia is global. And I like this quote from McGlock, who says that demography is not destiny. You can change this with education, with social circumstances, and the kind of things you eat. And those are interesting facts that we brought out from our study. There's lower dementia burden in the Yoruba. There are some differences in dementia risk in the two populations. These are vascular genetic. Resilience, which is the theme of this conference, is what exploring through clinical imaging, neurobanking, and various omic studies. And this is the reason why I'm excited about this conference, and we hope we're going to learn a lot. Well, these are the two teams, Indianapolis team, and then this is the Ibadan team. And we need to remember those who have died. Oshintokun started the study, and Kathleen Hall. And I'd like to thank McGlock and Valentina. Like they said, they are holding the brains in their hands with what they are doing. And we're looking forward to such exciting results from these studies. And these are the various funding things that we've talked about. The last one is the current study that we are doing, the supplement. And I'd like to thank you all for listening. Thank you so much.
Questions? Since you were so efficient. Okay.
All right, so that's fine. Yeah.
Any questions? And can I just ask people, since part of this is sort of a workshop-style format to get to know people, if anyone who asks the questions just identifies themselves by name and where they are and where they're coming from?
My name is Destiny. I came from Arizona, went to Arizona State University, and I'm doing research in autism. So my question is, do you think that the slave trade and slavery and the environmental effects of that over generations had an effect on people's genomics and different genetic predispositions to Alzheimer's disease and different things potentially?
The answer is yes or no. I think yes, because over time there has been genetic admixture, and so things have changed, and it's been calculated that about 80% of that has happened. The other thing is we're looking at various environmental things that may be dominant in determining disease risks, and so that's what I will say now. Thank you.
We still have time for any other questions?
So I'm Marco from the University of Iowa. I'm a fifth-year PhD student. I study cognitive reserve resilience and non-clinical.
You talk a little louder.
I study reserve resilience and cognitively intact older adults. My lab's interested in exercise and intervention. I have some cardiovascular stuff that I'm going to take later today. But my question is about that plot that you showed with APCA7 risk. That look like African Americans, so individuals who are in the U.S. show an interaction with that gene, but not from the Yoruba sample. Is that right? My interpretation of that plot?
No, the two groups, the two cohorts had interaction with cholesterol. They both did.
And they were the same effect?
Yes, about the same effect in the two populations, although the risk was higher in the African Americans than in the Yoruba.
Okay.
Yeah. Okay. Do we have time for a little more questions? Anyone else? Anything else? Any other questions? Going once? Okay. So thank you very much.
Thank you.
For coming so far.
Thank you.